Greetings from Amala. Let's continue the MCQ on microbial instrumentation and electrophoresis part 2. Which electrode is used as a reference in pH meter? Calomel electrode, silver chloride electrode, platinum electrode, glass electrode. The correct answer, Calomel electrode. What kind of calibration standard is used for pH meter? Buffers with defined pH value, conductivity solution, saline solution, glucose solution. The correct answer, buffers with defined pH values. In a hot air oven, what is the primary mode of heat transfer? Convection, conduction, radiation, convection and conduction. The correct answer, convection. Which parameter is critical to ensure proper sterilization in an autoclave? Humidity level, sterilization level, load, weight, UV exposure. The correct answer, sterilization time. Which biological materials are typically lyophilized for long-term storage? Proteins and cells, DNA and RNA, antibodies and enzymes, all of the above. The correct answer, all of the above. What is the advantage of using HEPA filter in a laminar flow cabinet? Increases air temperature, removes 99.97% of airborne particles, decreases pressure in the cabinet, absorb UV light. The correct answer, removes 99.97% of airborne particles. What is the function of a vacuum pump in a lyophilizer? Provide cooling, reduce air pressure to enable sublimation, circulate heated air, sterilize samples. The correct answer, reduce air pressure to enable sublimation. Which type of incubator is used to grow microorganism under carbon dioxide condition? Anaerobic incubator, CO2 incubator, refrigerated incubator, shaking incubator. The correct answer, CO2 incubator. Which material is suitable for sterilization using a hot air oven? Surgical instruments, culture media, plastic pipettes, gloves. The correct answer, surgical instruments. What is the purpose of UV lamp in a laminar flow cabinet? To detect contamination, to sterilize the work area, to improve airflow, to observe moisture. The correct answer, to sterilize the work area. Which type of sample is most suited for colony counting using a colony counter? Pure liquid culture, mixed bacterial colonies on agar plates, solid media without colonies, samples in petri plate with no visible growth. The correct answer, mixed bacterial colonies on agar plates. What is the primary use of thermocouples in instruments like bacterial incubators? Detect humidity, detect temperature, measure pH, provide sterilization. The correct answer, detect temperature. What is the key difference between STS page and native page? STS page separates based on charge, native page by size. STS page separates by size, native page by native charge. Native page uses detergent, STS page doesn't. STS page require DNA, native page require RNA. The correct answer, STS page separates by size, native page by native charge. What is the main application of agarose gel electrophoresis? Protein purification, RNA sequencing, DNA fragment analysis, antigen detection. The correct answer, DNA fragment analysis. Which component of a buffer system ensures pH stability during electrophoresis? Water, salt, tris, agarose. The correct answer, tris. What is the purpose of glycerol in loading dye for electrophoresis? Strains the sample, increases sample density, fluorescence under UV light, lower the pH of the buffer. The correct answer, increases sample density. In immunoelectrophoresis, what forms the visible precipitin lines? Protein aggregates, DNA strands, antigen-antibody complexes, buffer salts. The correct answer, antigen-antibody complexes. Which detector is typically used in capillary electrophoresis? Fluorescence detector, UV visible detector, mass spectrometer, all of the above. The correct answer, all of the above. Which electrophoretic technique is commonly used to separate hemoglobin variants? Agarose gel electrophoresis, paper electrophoresis, capillary electrophoresis, immunoelectrophoresis. The correct answer, paper electrophoresis. What is the purpose of reducing agents like beta mercapitoethanol in STS page? Stains protein, breaks disulfide bond in protein, maintaining buffer pH, polymerize the gel. Correct answer, breaks disulfide bond in protein. What is the function of a stacking gel in STS page? Separates protein by molecular weight, denatures protein, concentrates protein into sharp bands, bind to the dye molecules. The correct answer, concentrates protein into sharp bands. What is the key advantage of immunoelectrophoresis over gel electrophoresis? Faster result, higher specificity for antigen, lower cost, no need for buffer. The correct answer, higher specificity for antigen. What type of gel is used in capillary electrophoresis? Agarose gel, polyacrylamide gel, no gel is used, cellulose acetate gel. The correct answer, no gel is used. What is the migration direction of negatively charged molecules during electrophoresis? Toward the anode, toward the cathode, random direction, no migration. The correct answer, toward the anode. What is the advantage of silver staining over Comasi blue staining in STS page? Simpler procedure, higher sensitivity, non-toxic reagents, faster processing time. The correct answer, higher sensitivity. In capillary electrophoresis, what determines the rate of migration of analytes? Molecular size only, electric field and charge, sample volume, buffer viscosity only. The correct answer, electric field and charge. What is the function of bromophenol blue in STS page? Stains protein, act as a marker dye to monitor electrophoresis, denature proteins, maintains pH. The correct answer, act as a marker dye to monitor electrophoresis. What is the key difference between DNA and protein gels? DNA gels are stained with Comasi blue. Protein gels use agarose. DNA gels use polyacrylamide. DNA gels use agarose. Protein gels use polyacrylamide. DNA gels run horizontally. Protein gels vertically. The correct answer, DNA gels use agarose. Protein gel use polyacrylamide. Which of the following is used to detect RNA in electrophoresis? Comasi blue, malachite green, ethidium bromide, northern blood. The correct answer, northern blood. Which electrophoresis technique is widely used in forensic DNA analysis? Immunoelectrophoresis, SDS page, capillary electrophoresis, paper electrophoresis. The correct answer, capillary electrophoresis. Which instrument is essential for sterilizing media and equipment in a microbiology lab? Bacterial incubator, laminar flow cabinet, autoclave, hot air oven. The correct answer, autoclave. Which part of a pH meter detects hydrogen ion activity? Reference electrode, glass electrode, platinum electrode, salt bridge. Correct answer, glass electrode. What is the main advantage of a shaking incubator? Provide UV sterilization, maintaining sterile airflow, enhances oxygen availability for culture, removes excess moisture from media. Correct answer, enhances oxygen availability for cultures. What safety precaution must be followed when using an autoclave? Keep the door open during operation. Use only dry air cycles. Releases pressure before opening the lid. Operate a low temperature only. The correct answer, release pressure before opening the lid. What type of biological sample is best suited for lyophilization? Volatile liquids, heat sensitive materials, non-biological sample, highly hydrated gels. The correct answer, heat sensitive materials. Which environmental factor is controlled in a carbon dioxide incubator? Light sensitivity, humidity only, temperature and carbon dioxide levels, pressure and air circulation. The correct answer, temperature and carbon dioxide level. What does a colony counter primarily measures? Colony size, optical density, colony forming units, growth rate. The correct answer, colony forming units, CFUs. Which material cannot be sterilized in a hot air oven? Glassware, surgical tools, plastic pipettes, metal instruments. The correct answer, plastic pipettes. Which instrument is designed for high precision temperature control? Lyophilizer, autoclave, bacterial incubator, hot air oven. The correct answer, bacterial incubator. What is the role of ethanol in maintaining laminar flow cabinet sterility? Enhancing air filtration, removes humidity, acts as a surface disinfectant, neutralizes pH. The correct answer, act as a surface disinfectant. Thank you. Stay tuned for next video.